I'd now like to have a look at an overview of plant genetic transformation. And in this overview I'm going to have a look at an introduction to plant genetic transformation, the current status of GM crops and future trends and problems. I'm not going to cover the techniques of plant genetic transformation. That's going to be in the next screencast. So I want to talk about the potential of plant biotechnology, the uses of introduced novel genes, and traits that plant breeders would like in plants. There is a tension between the genes that are available and the traits that plant breeders would like in plants. Plant biotechnology as a whole has huge potential. We've already talked about micropropagation, but it's also possible to make what are called somatic hybrids or cybrids, where you can fuse the cytoplasm from one plant with a nucleus from another plant. This is very important because both the mitochondrion and the chloroplast actually do have their own genome. So it is possible to actually produce different kinds of plants by making somatic hybrids. A good example of this would be that cytoplasmic male sterility, which causes the pollen to abort, is widely used in corn, is actually mitochondrially inherited. So it's maternally inherited. The cytoplasm doesn't survive in pollen, and both chloroplasts and mitochondria are omitted from the pollen. So it means that mitochondrial and chloroplast traits have to be maternally inherited. By using anthiculture, it's possible to produce plants de novo from the pollen. And the pollen grains are haploid, so you end up with haploid plants. And again, these have a lot of uses in terms of uncovering recessive genes. The fermentation industry in plants has been widely researched, but it's not widely used because it is expensive. And finally, I want to talk today about introduction of novel genes into plants. And these have a number of different uses. You can research into gene functions by putting in antisense to knock out particular genes. You can get overexpression of one particular protein. So for example, human serum albumin can be expressed in plants, and you can end up with high levels of human serum albumin being formed, which can then be purified. So this is an example of molecular farming. Or what's been used most widely is crop improvement in a single step. In terms of molecular farming, a good example would be polyhydroxybutyrate. So plastics are currently produced from oil, and oil is a diminishing resource. But some bacteria produce this compound, polyhydroxybutyrate. So the genes for polyhydroxybutyrate production have been taken out of bacteria and put into plants. So it's possible to get appreciable level of polyhydroxybutyrate in plants. If you clone the genes for antibodies into plants, then you can get monoclonal antibodies being produced in plants. And you can produce different antibodies by doing different sexual crosses. I've already mentioned human serum albumin, but there are a wide variety of interleukins which are used medically that can be expressed, and also vaccines. So production of viruses in plants is due to, and animals, is due to production of viral coat protein genes first. So if you can interfere with this, you can interfere with virus production. So it may be possible, for example, to grow your own vaccines. Neurotransmitters Examples of this, so for example, leuenkephalin have been produced in oilseed rape. Oils have been modified to improve biodiesel. One of the problems with biodiesel, so biodiesel is basically diesel which is produced from biological sources, principally rapeseed oil, but you can improve the quality of that. Rapeseed oil has a relatively high gelling temperature, so you go out to your car on a winter's day and you find that your car won't start because the biodiesel in the tank has actually gelled. And it's a problem with all diesels, but particularly with biodiesels. But you can introduce, for example, change the ratio of double and single bonds in your biodiesel to affect the gelling temperature. And a company called Prodigene is now producing enzymes, oral vaccines and antibodies from maize seeds. One of the problems that you have in producing single proteins for molecular farming is that they get degraded in the cell. So what Prodigene have done is they've actually taken the promoter from this naturally expressed in maize seeds for a particular maize storage protein, and in that maize storage protein they've put a particular restriction site, so you can clone in an extra bit of DNA in order to produce your product. Then when you harvest your seeds, you recover the protein, and then you can nick out your particular product with specific proteases which have been also engineered into the multi-cloning site. So this is from Prodigene. So this reckon that the gene isolation is easy. The vector design includes organ-specific promoters, so they can get expression in the seed. They're using high-level promoters to get high-level expression, and the gene is actually contained. It's contained in the seed itself. I'll be talking about transformation later, but there are a number of different ways of getting genes into plants, and one of them is by blasting the genes in using a particle gun in a process called biolistics. I'll be talking about this in the next lecture. One of the things about monocots is that regeneration from a crop monocot is difficult. Now Prodigene have actually optimized the particular species and the variant of that particular species in order to get a working micropropagation protocol or regeneration protocol, so they can regenerate whole plants back from transformed cells. And one advantage of Prodigene is that they've optimized the growth, seed harvesting and downstream processing, and they are actually an agricultural company, and this requires strong agricultural and fermentation expertise which they have, so it's a selling point for Prodigene. But they are far from being the only company who's doing this. Okay, so what traits would plant breeders like to have in plants? Well, high primary productivity is one, high crop yield is another one, high nutritional quality is another one, adaptation to intercropping is one, and nitrogen fixation is one. These are problematical because they are complex multi-gene traits. When we're putting genes into plants, we typically put in one gene at a time, and ideally only one gene. So these traits on the left there are not really amenable to improvement through plant genetic manipulation. However, the traits on the right there, drought resistance, pest resistance, adaptation to mechanized farming, insensitivity to photoperiod and elimination of toxic compounds, can be done reasonably easily by the introduction of a specific transgene.